Assalamu alaikum everyone. My name is Sabrin Alam. My ID number is 2023100000164 from the Department of Computer Science and Engineering at Southeast University. Today I will present my research title, which name is Multiple Class Shrimp Disease Detection Using Deep Convolutional Neural Networks with Explainable AI. Here is the introduction and the background of the study in this research paper. Let me start with some background. Shrimp farming is also called shrimp aquaculture is one of the most important industries in the world. Millions of people depend on it for food and income, especially in these countries like China, India, Vietnam, and Indonesia. In fact, global shrimp production crossed 5 metric tons in 2023, according to the, and this is actually huge number. And the introduction part is shrimp cultivation is a significant of the world fishing industry. Shrimp cultivation is vital activity for the economic growth of many nations. Financial issues and losses are incurred because disease outbreaks annually. Let's moving to our third slide, which is research gap. In this research gap, we are actually see the different kind of research gap. The first one is limited multi-class detection. Most existing models focus on binary classification. Here is the binary means they have only two part, uh, and there is no different type of disease detection in there. AI models before this only answer one question: is this shrimp sick or healthy? Is limited multi-class detection that is called the binary classification. And but in this reality, shrimp can have different types of disease at the same time. No previous model can detect all disease types together in one system. And the second one is co-affection was ignored. Overlapping symptoms in dual infections, BZ, which means black gill, and WSSB, which mean white, uh, white, white shrimp disease. Uh, WSSB means white shrimp virus disease cause significant misclassification. Period work largely neglects these harder multi-pathological cases. The third one is lack of explainability. Most deep learning models in aquaculture act as black boxes. Black boxes, like most AI model works, they give you answer and cannot tell you why they give that answer. A farmer or a doctor cannot trust a system that does not explain itself. So this is the last research gap in here. And the second and the next slide is slide number 4, problem statement. How can we develop an accurate, robust, and interpretable deep learning system to simultaneously classify shrimp health into four classes: healthy, BG infected, WSSB infected, and BG with WSSB co-infected from image data alone? Here we can see the K challenges. The K challenges is the first one. Challenge is visual ambiguity. Black gill and white spot virus look like a similar in this photo. The symptoms overlap. So even a trained eye can get confused. That is called visual ambiguity. The second one is Data imbalance. Data imbalance, different number of images for each classes like healthy 403, busy 198, WSSB 328, busy with WSSB 220. And the third challenge is co-infection detection. Here we can see the when both diseases are present together, that symptom mix and each other. This is the hardest class to detect in this problem of statement. And the fourth one challenge is interpretability. Answer. It must show why it made that decision so that expert to trust and deploy them and use it. We next jump into our next slide, which is scope of research. Now, let me define the scope, meaning exactly what this research covers. Dataset here used the 1,149 raw shrimp images across four health classes. We are actually said that in this previous lecture, in these four classes is healthy, BG, WSSB, and BG with WSSB, which is co-infection under the standard aquaculture condition. And the second one is validation. To measure results are reliable. Here used five-fold cross validation, meaning trained and tested data 5 different times and different portion of data. Each fold run for 10 to 15 epochs. And the third one is explainability. Explainability here the use technique called gradient weighted class activation mapping applied to verify biological relevant feature learning. And the fourth one is models. Here using three type of deep learning model. The first one is inception V3, the second one is exception, and the and novel dual fusion model are third number. In these five number of matrices, it measures the performance using accuracy, precision, F1 score, TPR, TNR, FPR, FNR, and the confusion matrix of comprehensive evaluation. The last one is limitation. The dataset only covers two diseases, uses statics photos only, and has not been tested on mobile or field devices. And these are areas of future improvement. Our next slide is summarized literature review. Let me quick walk you through that what other research have done before and what they missed. Look at this table. Here, Bio at AL 2019 used Raspberry Pi OpenCV plus Niblex algorithm. And they are camera to detect one specific device. YOLO had virus, but it was binary only and only worked that one embedded design. Here is the research gap, binary and embedded only. Deong Trang et al. 2020. They are using this method is transfer learning CNN and the K finding high classification accuracy for shrimp descent from the images. Here is the research gap. We identified that no co-infection classes. Korawit et al. 2024 transfer learning CNN used the method is transfer learning CNN and the K findings is detect physical defects in the shrimp processing, head loss and tail cuts, and we identify the gap is physical defects only, not disease. NUN EAT AL 2022. In this reference uses our CNN method and field images, Mekong Delta. And the key finding is early detection tool for farmers, one side decision support, and the gap identified with binary and limited classes. XAI in aquaculture. Here we are using this method, explainable AI, and the key finding enhance transparency of AI decision for aquaculture expert, rarely applied to shrimp disease. We are moving to our next slide, which is methodology. This slide is very important because methodology explain us what actually working on this paper. Now let me explain how build the system. You can see here are you can see here are six steps. The step number one is dataset. Here is 1149 images with four classes. In this all data gathering to preserve this data set, and therefore the next step is pre-processing. Here we are using the dataset and resize 224 into 224 pixels, and that normalize into this 0 and 1. And the third one is augmentation, because that is the limited images that is the limited images here is used especially for busy class. Here is the create extra training data by flipping, rotating, zooming, and translating, shifting the existing image. It helps the model learn better. And the fourth one is model training. This is the most important part in this paper. Here three model are uses: inception, B3 exception, and dual exception uses in here. And here is also step 5, which actually I made mistake, but 5 and 6 step are combined in there. So here is great CAM visual model decision. Now most contribution, the dual exception model, image having two expert doctors looking at the same stream photo at the different details, then they combine what they both noticed. That is actually working on this explainable AI. So this is exactly what dual exception does. It has two parallel exception branches. Each branches learn different feature from the same image. Then the feature are combined and the passed through final layer and give the disease prediction. This is more powerful than using just one model because capture more information from image. So we are jump into our next slide, which is result model performance comparison. Here we can see the three models in use in this paper. We already said that there are three models in here, which is inception V3. We are already seeing that this is the accuracy range of 69% to 73.37%, and the best F1 BZ 0.59 and best F1 WSSB with the BZ is 0.84, which is not better than others. And the second one is the exception, which accuracy range is 70.53% to 76.33% accuracy, and the best F1 BZ 0.65 and the best F1 WSSB with BZ 0.77, which is better than compared to Inception V3. This is little bit better. And the last one is dual exception, which is most better than all of these two models. Here is 77.30% to 87.50% accuracy range, and best F1 BZ remain unchanged compared to exception. And the Best F1 SSB with BG is very high performance 0.91. So we are actually see the K observation. Real exception obviously here is better performance to others and achieve it up to 87.5% accuracy fold 2. BG class FNR reduced to 0.08 and critical for DJs screening. WSSB with PG co-infection F1 up to 0.91 versus 0.31 in exception fold 4. Consistent performance across all 5 folds. So we are jump into our next slide, which is explainable AI grade say CAM visualization. So here is the visualization of this presentation. This technique creates a heat map on top of the shrimp image. The bright areas show where the AI looking and when it made its decision. So let's explain the four features in here. The first one is healthy shrimp. The heat map was spread lightly across the whole body. This makes sense because AI was checking there are no unusual spots or dark areas anywhere and showed strong activation around the chest, gill and chest diseases, and therefore this is the dark areas anywhere. And the second one is black gill bg. Here is high activation around the gill and cephalothorax region, precisely where gill darkening and decoloration are expected pathologically. And the third one is WSSV, white spot virus. White spot virus, the heat map focused on the abdominal region and the shrimp belly. This is biological accurate because white spot virus cause white patches on the body and tail. And the last one is BZ plus WSSB, which called co-infection. Both are occurring in the same time. So the heat map activate in both the gill area and the belly. The model correctly identified symptoms in two different body regions in the same time. The provides of the AI not guessing randomly, it is learning real biologically pattern. This is the explainability that makes the model most trustworthy of real world use and situation. And my next slide is conclusion. Here is the conclusion. We are step by step summarize this conclusion. Here are 5 steps in there. I actually combined all of these without numbering. So let's explain the 5 points in here. The first one is dual exception outperform inception V3 and exception on all matrices across 5-fold cross validation. The second one is achieve up to 87.5% accuracy with high F1 scores even for difficult co-infection plus. WSSB with BG F1 is 0.91. The third one is significantly reduce false negative critical for real world disease screening application. Grade CAM confirm biologically meaningful feature learning in gill and abdominal region. And the fifth one is provides an interpretable, accurate framework suitable for agriculture, surveillance, and development. Here is our next slide. We actually working on this in our future work for this paper. We can see the expanded dataset include more diverse environment, lighting condition, shrimp species, and the additional diseases class to improve generalizability. Here they are actually want to know add more shrimp species, different lighting condition, and more disease type make the model work is more real world solution. The second one is edge device development. Optimize and deploy the model on smartphones or embedded robotics for real-time field level disease detection by farmers. They are actually want to say optimize the model so it can run on a smartphone small device that a farmer can carry easily and fish pond and it use immediately. And the third one is temporal data fusion. Combine long-term behavioral observation data with image feature for richer multimodal shrimp health assessment. They are actually want to say that right now the model only looks on the one looks at one photo. In the future, we can also use video or behavioral data like how the shrimp moves over time to make even better prediction. And the last one is more AI techniques. Explores SHAPLIME and the attention map visualization alongside great CAM for richer model interpretability. They are actually want to say that explore shape and line techniques alongside great CAM for even deeper model explanation. In this all of work, we are actually working on our in future work for this paper. This is very important for our paper. And the next slide is references. Here are 11 reference papers that support this work. All references follow paper academic citation format. I'm happy to discuss any kind of this paper in there are questions. And the last slide is now this is bring me to the end of my presentation. Thank you so much for your time and attention. Now I am open for any question and any discussion. Please feel free to ask me any question about this research models and result. I will do my best to answer all of this question. And thank you for everyone.